Welcome to the next installment of this sort of seminar mini-series that I'm doing looking at machine learning and looking at some examples in biological sciences. So this is the third installment. There will be two more after this, so we're right in the middle. This time we're going to be looking at, again, using sequences like we did last time, but a slightly more complex approach to deal with some of the things that we couldn't deal with previously. So things like different sequence lengths, and just generally if we want slightly more complexity in our model to allow us to be a bit more flexible in our approach. I have changed the paper that we're basing this off of compared to the one that I promised you previously. So apologies if you were super excited for that specific paper, but I think this has some more considerations that are helpful to think about when we're thinking about the use of machine learning. So hopefully you will also agree that it's a good switch. So what we're going to cover, again, this looks very similar to what we've looked at previously. I'll start off with a very quick introduction to machine learning, just in case anyone hasn't made it to any of the previous seminars. I'll then go through a quick introduction to the paper, looking at the datasets used and the kind of questions that they're trying to answer. Then go through some of the pre-processing, so their approaches around data tidying or feature extraction, which will be a big thing for this one. And then I'll go through the model, how it works and the different stages to it, and some validation that they did, and finally some sort of further exploration that they did using the results from the model to show some examples of applications. I will, at the end, very briefly have a side note on how to run part of the model in Tidy Models. But there will not be a practical section in this one to give more time to talk through the model, and also because it's slightly more complex, it's slightly harder to do a proper demonstration of, but hopefully it will still be useful to think through the concepts and how these things work. So, what is machine learning? Some of you may have seen this 2 times already, so I will go through it quite quickly. In a usual data analysis workflow, we will have sort of 4 key stages. So we will have collection, where we will get some raw data from whatever kind of experiment or is that we're doing. We will then do some kind of preparation. So we will visualize and explore our data, probably do some filtering or some cleaning based on this and cycle through those so we understand what's happening in our data and what it contains. Usually we would then formalize anything that we've seen using statistics, and report our outcome in the form of some kind of statistical significance usually, and test statistic potentially associated with that. When we're doing machine learning, instead of statistics, we are doing some kind of modeling as our formalization. That might be interesting in itself to show that we can produce a model based on our data, and so we might just report that, or we might then want to use our model to predict things. And this is often why we end up using machine learning, is to make predictions on new data based on what we've seen in our training data. So the steps that we follow, we always have to start off with data where we have samples where the outcome that we want to predict is known. We can then pick a model, or pick a method for our model and build said model, and evaluate it and refine it and reevaluate in as many loops as we may require. And then we can make predictions based on this where the outcome is not known. And we may also want to test those predictions as well going forwards to make sure that what we're seeing is realistic and it's still working well for us. So again, some key differences between our models that can help us to decide what to use. Firstly, our outcome types. So we may have regression models if we want a quantitative prediction, classification model if we want a categorical prediction. Some model types can be both. And also, depending on our input type, may also depend what model we choose or may influence which model we choose. So some models require all of their variables to be numeric. So we may need to convert any categorical values to numbers. Some models also have expected behavior of their input data. So something require it to be, or assume that your data will be normally distributed. Some don't care what your data looks like so much. And there's also variation in the number of viable measures, so in the size of your data set and the number of predictors that you may have in your model. As we will see today, some of them can have quite large data sets. Some don't work so well with really large data sets, and so that can be another consideration of which model type that you use. And just as a reminder, some of the machine learning models that we can use, we've already seen linear regression. That was the first one we looked at. Last time we looked at support vector machines. This time we are going to look at neural networks, but actually sort of neural networks plus. So we'll have some steps before we actually get into our neural network, as we will see shortly. So this is the paper we're going to look at. We're looking at predicting the sequence specificities of DNA and RNA binding proteins by using this mystical term of deep learning. So we will go through what this is actually doing, so that it's hopefully a bit less mystical by the end. So what is the data? Essentially, we're trying to identify sequence specificities of DNA and RNA binding proteins. So therefore, the input for our model when we're training it is going to be a set of sequences and some associated experimentally determined binding score. The binding scores can either be measurements of the strength of binding, or they can be binary class labels in this case. So either has a binding or does not bind. So that means we can either have regression or classification models in this case. The other difference from last time is that this time our sequences can have varying lengths. So in the paper, they had sequences between like 14 to 101 nucleotides in length. So they can vary, and the model is able to cope with that based on what we are actually entering into the neural network. They used a large amount of data in this paper. So they had 12 terabases of sequence data. They used thousands of different public repositories or public data sets that all gave different values for protein binding to either DNA or RNA. And also just to note where there were data sets that didn't have any background sequences. So for example, with the ChIP-seq data, you just get the essentially positive ones, so the ones that have bound. They used something called dinucleotide preserving shuffle on the original sequences. So on the positive sequences, they essentially kept pairs of nucleotides together and then randomized the sequence, maintaining that pairing between each two sets of nucleotides. The reason they did this is to maintain some of the structure that you might see in sort of real nucleotide data, real like DNA or whatever. So there is usually some kind of structure, so you're more likely to see particular bases next to each other. And so the hope was that they would be slightly more realistic background sequences than just completely randomizing. So, an overview of what this paper sort of very broadly looked like. They started off with these high throughput experiments that they combined together into a very large scale data set that they then passed through massively parallel deep learning So they did automatic model training, they had some kind of neural network going on, and they actually, in this case, built quite a large number of different models. So this isn't just one single model that they built a whole load for different transcription factors and RNA binding proteins and things like that. With the hope that they could meet some community needs, so looking at things like gene regulation, precision medicine, so seeing if any like single nucleotide variant has an impact on the binding, and also just looking at detecting binding sites themselves. So we're going to focus on this step in the middle, which they have nicely put into a diagram again. So this is an overview of the model, which we are going to talk through today. So you start off with some kind of batch of sequences that we're going to pass into our model. You can see there's quite a number of steps that we have going on, and that will give us some kind of output that we can compare to our target to see how well our model is working. We also have training data and testing data, as we have seen previously, so we don't test based on the same data as we trained, so that we can see how well it is working. But we will go through all of these steps in much more detail. So they called their model the deep bind model, and it's based on deep convolutional neural networks. So the reason they went for this is because they can have different lengths of sequences as inputs. They could learn from a very large number of sequences, and they used parallel implementation so that they could use a GPU to help speed up the process. They also found, or this allows them to apply it to different kinds of data, so microarray and sequencing data, and also cope with different technologies and things without necessarily having to overly correct for the biases associated with those technologies. And the reason for this is this kind of approach tolerates a moderate amount of noise in the data. And it also tolerates a moderate amount of mislabeling and things like that. So it means they didn't have to go through and manually curate every single tiny detail in the many, many, many thousands of sequencing data that they used in this. It also meant that we can discover new patterns, even when the location of the patterns within the sequences are unknown. So if the binding motif is right at the start of the sequence, it won't matter. And it won't matter if it's right at the end or in the middle. You don't have to say, and it doesn't have to be consistent where the binding motif actually is. And they also have set it up so that you can train predictive models fully automatically, so you don't have to go through sort of manual stages of optimizing the parameters of your model. And finally, they use the output so the predicted specificities could be explored and visualized in different ways that they have put together. So, as always, the first step is going to be looking at some pre-processing. So last time, we used something called one-hot encoding, where we took a sequence like ATGG here, and we used 4 ones and zeros to define which base was which, and essentially put them all along in a line. So we had one column or 4 columns for each base in our sequence here. So A ended up being 1, 0, 0, 0, T was 0, 0, 0, 1, G was 0, 1, 0, 0, and then repeat that at the end for ATGG. This time, we're going to convert our sequence to what they have called padded 1 of 4 matrix, and then we'll use that as our input to our convolutional neural network. So what this looks like is this. So we still have the same coding for each of our bases. So our A still becomes a 1, 0, 0, 0, T still becomes 0, 0, 0, 1, G still becomes 0, 0, 1, 0, and repeats. But this time we're keeping it in a matrix rather than putting it all along a single line. And we're also adding in some extra padding at the end, or at the start and the end of the sequence, which we will see how that helps later. But the amount of padding I've shown here is just like 2 rows. But the actual amount used in the model will depend on the length of the motif that we are searching for, which again, we will come back to a moment in a moment what that actually means. So convolution neural networks are mostly used for imaging data. So the vast majority of the time when you see these, it will likely be used on imaging data. In this case, we've adapted it for use on sequences. But the general idea is that you are able to take a large amount of fairly sparse data, so where we had quite a lot of zeros in our matrix here, and reduce that down to have a smaller input to our neural network or our model at the end. It also allows for small movements in the data, so it's less sensitive to noise, or having the same positioning of the sequence of interest, as I was saying earlier. And the other really cool thing about it is it can take into account correlations between adjacent points. So it will look at a few different bases together rather than just looking at a single base and calculating based on that, which is obviously useful in imaging data and also useful with our sequence data here. So we're going to have 4 stages to this. These stages are convolution, rectification, pooling, and then finally a neural network. And so we will go through what each of these are. So for convolution, we take our sequence or our converted sequence and apply something called a filter. In the paper, and for this, I will mostly call it a motif, but if you look more generally at CNNs, it may also be called a filter. So we'll use those slightly interchangeably, but they mean the same thing in this instance. In this little example, we are going to use a filter that is of length 3. So we essentially have 3 bases being included if we think each of these rows is a base in our sequence. And what we're going to do is we're going to use our filter to run through our converted sequence over here. So if we look at the first 3 bases here, so we have a same size sort of section as our filter. We then compare this part of our sequence to our filter. So we're going to do the sum of the products of each of these. So we will do 0.25 times 1 plus 0.25 times 0 plus 0.25 times 0 plus 0.25 times 0, and repeat that for each row. And then add all of those together. And for this first part, we then get a sum of 0.5. We're then going to move down 1 row and do the exact same thing, but for this next section of our converted sequence. So our first row will again be 0.25 times 1 plus 0.25 times 0 3 times. But our next 2 rows, we now have actual bases. So we do, again, 1 times 0, 0 times 0, 0 times 1, etc. Add them all together and get a value here of 0.25. And then we move down again. And this time we actually have some overlapping ones. So we can see this one overlaps with this one and this one overlaps with this one. So this time we add it all up together and get a value of 2. And etc., etc. We run down the length of our sequence, and we can produce something called a feature map. So this is just the first 3 were added up to get 0.5, the next one was 0.25, this one was 2. Keep going down, you get values of 2, 1.25, and 0.5 here. In this example, we have 1 motif, and so we have 1 column in our feature map. In the paper, they actually included multiple motifs for each sequence, so usually 16. And so therefore they had 16 columns in their feature map, one for each filter that they've run through their sequence. So convolution, we take a sequence and we apply a filter, which in this case is a motif, and it outputs a feature map where we have one column per motif. The feature map shows how similar our sequence is to the filter at any given point in our sequence. And in the paper, they include multiple motifs, so we have 16 columns, usually in our feature map. The other thing to note is that we also vary the length of the motifs. So in the paper, they do not use motifs of length 3, but this depends on the length of the binding motif that they're trying to detect. And all of this can be optimized while we're running through the training of the model as well. We don't have to specify the motifs, so we don't have to have prior knowledge on what we think the binding site will be, or the binding motif will be. The model will figure that out for us as we train it using our training data. So the next step is rectification. This takes our feature map as an input. And in this case, they use something called a rectified motif scan, using a nonlinear transformation through a rectified linear unit or ReLU layer. Essentially, this adds a coefficient, which we're calling beta here, for each motif that acts as an activation threshold. So if our values fall below this threshold, then they will be reduced to 0. So if we have a coefficient of 0.5, then anything that is 0.5 or below will just become 0, and anything that is above 0.5 will stay in the feature map after rectification. This ReLU layer or function looks like this. So you have basically anything that's below your threshold. So say this point here is 0.5, gets converted to a 0. Anything that's above just remains the same. I'm showing you this now because we will see it again later when we come to our neural network. The other thing to notice with this is the padding that we added at either end has now essentially just been removed because these have all just ended up as zeros. So this is where the padding doesn't necessarily add too much noise, but it does mean that, for example, this point down here where it has a 1.25, so there was actually decent matching with our filter for the sort of last 2 real bases in our data, we can still see that there is overlap here in our feature map after rectification. So it means you scan over each base the same number of times rather than missing out the first ones on your first sort of run through of the filter. So that's why you add in the padding. The next step is pooling. So this time we're applying another filter, but this time it's not going to overlap. So before we would just take it one step at a time, but this time we're just going to take, in this example, our feature map in 2 chunks, and we're going to take the maximum value within each of these filters, which would give us, in this case, a not super exciting max pooling output of 2 and 2. There is also another approach, which is taking the average value. As you would expect, we're taking the average of the first 3, the average of the last 3. So we get the mean pooling output would be 0.667 and 1.083. This is a slight simplification again, obviously. In the paper, because they have 16 different motifs, they actually just do pooling for each of those motifs. So rather than splitting it in half, it would take overall for this the value of 2 as the max pooling, and then you would have 15 other motifs, so you would have 16 input values for our model in the next step. Actually, in the paper, they use both max and average. They found that for DNA binding, the max was sufficient, so they just used the max. But for RNA binding, sorry, it was more complex. So they used both the max pooling and the mean pooling and used both of those as an input to the model. This is the step that means that it doesn't matter what length the original sequence was. So no matter how many values we had in our feature map, it is just summarized to one value. And so you always end up with the same sized output that can be used to your model no matter how long the sequence was. And it's this kind of summarization that also means that we're not too sensitive to noise because we're looking at either max or average values. So if you have some slight differences, it won't matter. And it also clearly doesn't matter whereabouts in the sequence you're matching this motif, because if this 2 was down at the bottom, it would still be the max value and therefore would still go forwards here. So now we have our pooled data, we can use it as an input to our neural network. So what does a neural network look like? In this case, I'm now expanded, so we have 5 different motifs, and we have the pooled value for each of these. The pooled values for our motifs become the input layer to our neural network. So neural networks are made up of nodes, which are these circles here. So we have 1 node per motif in this case. And layers, which is a collection of nodes. In this case, this is our input layer. So we have, in this case, 5 different nodes in our first layer. We will also have an output layer. In this case, I'm choosing the example of classification because it's easier to visualize. So our output that we want to predict is either whether it is binding or not binding. And we will also have some hidden layers that will be found in between our input and our output. And all of our nodes, which are all of our circles here, will be connected to each other. So from the input motif, all will be connected to all of the nodes in the hidden layers, which will also all be connected to the output layers. And we will see how they are connected in a minute. The thing to note with this, as a visualization, I'm including 2 hidden layers. In the paper, they actually only had 1 or even 0 hidden layers. Because of the complexities that came before this, they found that sometimes the hidden layers overfit the data. But essentially the number of hidden layers just allow or gives you more complexity in the relationship between your input and your output nodes. So if you have no hidden layers, you can look at a linear relationship between your motifs and the binding or not binding. If you have 1 hidden layer, you can look at nonlinear relationships. If you have 2 hidden layers, you can look at sort of nonlinear relationships with different inflections and things in your data. So you can model really complex relationships by having 2 or more hidden layers. In this case, they did not find that that was required. They actually only have a maximum of 1 hidden layer. So, as I said, neural networks. We have nodes and layers. Each node actually has an associated value within it. So our input layer, in this case, is the value that was outputted from our pooling stage. And these values here will be connected to the nodes in the next layer by some kind of mathematical relationship. So these lines here are essentially some mathematical transformation, which we'll see in a minute, that are associated with different weights and biases to calculate the next layer. And they are also passed through something called an activation function, which in this case is this ReLU, this rectified linear unit that we saw earlier. Although this time, instead of running to 0 until 0.5 in our example, it runs at 0 until we hit a positive value. So what does this actually look like? So say we have these three nodes here, and we want to calculate one of the nodes in the next layer for simplicity. We will have a number of weights associated with each individual connection of our input layer, and also something called a bias, which will be applied to each of these before we can calculate our next node value. So the first thing we're going to do is do the sum of our activation times our weight. So our activation here is the value inside our node. So in this case, we would do 2 times by 0.5, 1 times by minus 3.1, and 0.5 times by 0.6. So if we add all of those together, we get minus 1.8. And then we can add our bias here, which in this case is 1.5, and we get a value of minus 0.3. So we're going to pass that into our ReLU activation function, which will output whatever is larger, either 0 or the input value. So in this case, we have a negative value, so that will fall. If we can imagine this is a graph, it's a super highly detailed and impressive graph here, but this is a graph, this is our function here. Our value of minus 0.3 falls here, and so we're going to end up with a value of 0 for this node here. These values are all calculated when we train our model. So when we train a neural network, it will calculate the weights and biases as it's optimizing through the training rounds. Also, just a note, for those of you who have been on the machine learning course, you may remember that the activation values in that were set between 0 and 1. This is due to the different activation functions that we were using in either of these. So at that point, we were using sigma functions, which keeps it between 0 and 1. In this case, we just want to keep it to only be positive values, so we remove all of the negative values using this ReLU activation function. So how do we actually go about training our network? Well, the answer is we don't. Our model does it in the background, but how does our model go about training our network? So we can use something called backpropagation. So this is, again, just a small illustrative example. But we have, say, these activation values on the left here of our hidden layer nodes going to our output nodes, which in this case are either binding or not binding. So if we are trying to predict a single sequence that we've run through here, that does have a binding site, so we want to predict that it's binding, we can see here that actually the larger value in this case is in no binding, which would mean it would be predicted to not have a binding site. We want it to be predicted as binding though, because we know from our input that it does have a binding site. So we essentially want this to be higher and this to be lower. So how would we go about this? We can either increase our positive weights or decrease our negative weights. So we've again just simplified the mathematical relationship between our nodes. So the thicker lines here are larger weights. The green ones are positive, the orange ones are negative. So this is a strong positive relationship between this node here and this node here. And this is a strong negative relationship between this node here and this node here. Obviously, because we're timesing our original value by our weights, the strength of this relationship depends on both the sort of thickness of this line in this case, but also the initial starting values over here. So we can see these 0.1s, even though they have really strong linkages here, they're not really going to have that much of an input because we're starting off with a small number, whereas this is going to have a really large impact because it has a strong weight, a large weight, and also a large starting value. So if we want to increase this, we can increase the positive weights that are tied to high activation values. And we can also decrease the negative weights, but again, we're more interested in those that are tied to high activations upstream. So what we would want to do is probably make this one larger and this one smaller, so we have less of a positive impact on this no binding here. We're probably not going to care so much about these because they're tied to 0.1 and so therefore not going to have that much of an impact. This is done obviously not just for a single sample, but across all of our samples, or actually a subset of our samples, which is then done iteratively and adjusted as we go through our training. There are different ways to optimize this as we go through our training steps. I don't have time to go through all of that in a great amount of detail, but in this paper they used something called mini-batch stochastic gradient descent, or SGD. The mini-batch part of this means that they optimized based on subsets of the data rather than the whole data set itself. And the reason why they did this was one, to avoid overfitting, but also mostly two, because it runs a lot quicker when you're only trying to optimize on a subset rather than on the entire incredibly large training data set that you might be using. Another thing that they did to try to avoid overfitting in their model, which can be a very big issue when you're training on particularly very large data sets, is they used something called dropout regularization. This essentially just randomly drops a certain percentage of your nodes during training. And this is implemented per layer, obviously not in the output layer, but can be in the input and also in the hidden layers. It also is worth noting that the remaining active neurons or nodes that we have in our model are scaled up in relation to the amount of nodes that are lost. So if you lose 50% of your nodes, then you will scale up your other ones by sort of twofold to maintain a sort of similar overall level, essentially. And this is really useful to reduce the over-reliance on very specific connections between your nodes or your neurons. So if you use everything all of the time, you might find that there is one specific connection that becomes really important, but actually that might not be sort of generalizable. It might be overfitting to your data, so it's trying to avoid that. So what does this actually look like? We can go back to our original sort of structure of our neural network. Essentially, all we're going to do is on one training run, we're going to lose, say, one of these and maybe a couple of these in this hidden layer, and also increase the connections between these existing nodes. And then in another training iteration, we might lose some different ones. And again, we will sort of average out those relationships to try and build an overall model that tries to sort of summarize the relationships rather than just memorizing the training data itself. So that was a talk through hopefully all of the first part of this model overview that looked particularly scary to start with, but hopefully we've gone through this convolve stage, rectifying, pooling, and going through our neural network. We've also talked a little bit about training and what happens during the training stage of our neural network. But what we haven't talked about at all is this bottom left-hand side, which is calibration. And this is one of the cool things they've done in this model. So it's also called hyperparameter search, but it's essentially where we're tuning the model parameters. So once we get to these really large and complex machine learning models, there are a whole load of different model parameters that we can change and play around with. And if we're trying to do that manually, it gets very complicated and can have a really large impact on the overall success of our model. So instead of trying to do it manually, in the paper they have built a way to optimize all of these while calibrating their model. There are a whole load of different parameters that they calibrate during this. So something called the learning rate, which essentially changes how large the steps are when we're changing the weights and the biases in our neural network. Something called the momentum. This is something that essentially speeds up the learning of our model by saying if something is gradually getting larger each time we're running a training iteration, probably you can increase it by a larger amount next time because it seems to be going in that general direction. The mini batch size, which is what I was talking about previously, where we did the mini batch SVG. This just changes the size of the training subset that we use for optimization of our neural network during the training. Also the number of learning steps. So as we're going through and training our model, we might want to stop it slightly earlier than when it has completely optimized all of our weights and biases to try to reduce the overfitting of our model. The dropout probability is the proportion of no's that we drop during our dropout regularization. And also another calibration is whether or not to include a hidden layer. So the options are either include one or have none. And in the model that they built, they do 30 different calibration options with different combinations of these different parameters or different values of combinations of these different parameters to help determine the weight or the level of regularization. And regularization is essentially what we do to try to avoid overfitting. So each calibration trial uses a different set of calibration parameters, as I said, and they figure out the optimum calibration based on calculating an average AUC or area under the receiver operator curve if it's a classification, or the mean squared error if it's a regression, from a threefold cross-validation. So we can see down here, this means they split their training data into 3 subsections, and each time they use 2 of those sections to train and 1 to validate, and then they will calculate an average sort of measure of how good the model is, depending on whether it's classification or regression, and pick the one that ends up with the best, in this case, average validation AUC. So in this example, they had 1 to 30 down here, and the second trial of the calibration produced an AUC of 0.95, and so they used that going forwards into their proper training of their neural network down here. This is a very computationally intense process. If you have a very complex model with a large amount of data, apparently it could take up to weeks of computation to find the optimal parameters of this calibration stage. And so this is the main point at which they implemented their GPU acceleration process to reduce some of the time that this particular stage takes. So in the end, they built quite a large number of different models using this. So they outputted 927 of these DeepBind models that represented 538 different transcription factors and 194 different RNA binding proteins. Also, as a slight side note, when they had double-stranded DNA as an input, they evaluated the score for both directions of the double strand, and used the maximum of the 2 scores to produce a final prediction. When they did this, they used the exact same model versions, so they had like the same node dropouts when they were doing the regularization and things like that. Because they had so many different kinds of models, There are also so many different types of evaluation that they did depending on the model and the output. So I'm just going to talk through one or two of those. And they also extracted all of their trained models and put them into an online repository, as well as some of the details of how to run these models yourself if you wanted to train a model for a different transcription factor or whatever it is that you are interested in predicting binding for. So one of the ways they evaluated their DNA binding protein specificity is using some data from something called the DREAM5 TF-DNA Motif Recognition Challenge, which was essentially one of these challenges where they got some teams together to try and produce the best model to predict transcription factor binding, essentially. So it contained 86, I don't know where 20 came from, 86 different mouse transcription factors, measured using 2 different array designs. And so different teams trained their models based on the probe intensities using one array design and then predicted the intensities on the other one. They evaluated it using a sort of combined score, which they called the DREAM5 PBM score, which was based on the Pearson correlation between the predicted and the actual probe intensities, and also the AUC, which was calculated by just setting high intensity probes as positives and everything else as negatives. And they combined those across 66 of the transcription factors. So essentially, the authors trained or tested the DeepBind model under the exact same conditions as they did in this challenge and found that it performed better than any of the others that were produced. So a comparison against other models that were trying to do the same thing. And also within this DREAM5 challenge, they had a section looking at how well the in vivo transcription factor predicted in vivo values. And again, the DeepBind model worked very well for this in comparison to the others as well. So that was a short bit on validation. They did also look at some sort of further exploration of where this can be used. And there is a whole load more detail in the paper. So if you are interested in any of this or any of the other approaches, feel free to have a look at that. But one thing that they did that I think is quite cool is they identified and visualized potentially damaging genetic variants. So looked at variants in the binding sites that might alter the gene expression patterns. And so they developed this mutation map visualization, which is what this down here is, to try and illustrate the effect of different point mutations within a sequence on the binding affinity of the protein that they are trying to predict the binding for. So you can see in these, the really large letters are the bases that have a really strong influence on the binding of the protein. And the heat map underneath those shows the influence that a change in that base would have on the binding of the protein. So for example, if we take this T here, we can see that if it gets changed to any other base, you're going to have a massive decrease in the predicted binding of this protein here. And also you can see, hopefully, the little stars in there are mutations that have been seen in patients that do have some kind of phenotype associated with something to do with that protein of interest. So again, if you're interested, you can look up more details, but I think that's quite a cool output and shows that it does potentially have more widely applicable outputs as well. Okay, so I said I'm not going to go through a practical demonstration of this because I absolutely do not have time, and also it would be far too complicated. But just a slight note on tidy models. So as I said previously, tidy models is really nice. It provides a consistent interface to prepare our data and construct our models and also evaluate the results from our models by sort of talking to different packages in the background that actually do the modeling. But it means we can switch between them without too much code changes, and we can try different model types and play around with our data in different ways. So just as a slight example of how we might do the last step in our modeling here. So the neural network in tidy models can be done using the engine, which is the sort of background package that it uses, called Brule. And as we might expect, this model has a whole load of different parameters that we can specify. There are more than the ones that I've included here, but just to very quickly go through some of the relevant ones that we've seen today during this talk. So this epochs is an important one. That's the number of passes through the training data that we do. We've got the number of nodes in the hidden layers. We've got this activation parameter, which is the activation function. So in this case, this would be the ReLU. So this is where it takes out all the negatives and keeps the positives. We've got dropout, so we can specify the proportion of nodes to drop during our dropout regularization. And also the learning rate, which is how large the changes in our weights and biases are when we're training. Our optimization method, which I mentioned briefly, which was this stochastic gradient descent, or SGD. And also the momentum that I again mentioned briefly, which is how much to scale our learning rate or our optimization based on things always going in a similar direction. We can possibly speed it up a bit. And finally, batch size, which is again the proportion of our training that we use during this optimization step when we are training our neural network. So just to say all of the things that we've mentioned, you can set up and tune as parameters in tidy models as well. And as this is a neural network, we do have some pre-processing requirements, which are all covered by the pre-processing that we did initially. But we can't have any factor or categorical predictors in neural net, so they would have to be numeric values, which they were after our convolution and pooling and things. And everything should have a similar scale, which again they are, because they're all sort of coming from the same data type. So that's not too much of a concern. So yeah, we can run this neural network in tidy models if we so wish, and you can also go through some of the tuning stages, although it may not be quite as automated initially as it is based on the model included in the paper. Okay, so that was a quick, not so quick, a go through of convolution neural networks, using them for predicting DNA and RNA-binding proteins, or the specificities of DNA and RNA-binding proteins, I should say. This has been the third in this series. There are two more. So next time, in 2 weeks, we'll look at interpretable models using random forests, looking at its application to the biology of aging, which I think should be an interesting one. And then the last one we'll look at is imaging data, so looking at some more considerations of feature extraction and things when related to imaging data as well. So thank you all for coming. I will see you next time.